Yes, hello everyone. I'm going to give like a relatively a basic overview of what it is to kind of sequence these things. So you've seen a lot of talks around what the virus is, how it works, and how some of the information is being used with public health. So I'll show how we are doing this kind of the thing at the Garvan. And I just want to point out straight away that we're working with Rowena Ball and Bill Rollinson, and we couldn't do any of this without them. So thanks very much for all that kind of collaboration. Now, can I move my, yes. Okay, so if you're not aware, because you've been living under a rock, this is how Nanopore sequencing works. You essentially have this dongle we call a MinION, although they have larger devices, and this is the replaceable flow cell part. You have a bunch of Nanopore embedded in an array. You have about 2048 of them, and they can thread DNA or RNA through this. You get electrical signal, and then they convert that into your bases. So the great thing about this is that it's relatively quick, real time, can work in the field. So, and it's quite quick to set up as well. You can pretty much just pick one up and carry it into a lab and have it up and running in, under an hour. I've even set up one of the larger machines in, I think, 3 hours or something. So, really accessible and interesting devices. So the biggest thing that's happened with Nanopore and the analysis and quick turnaround of the bioinformatic pipelines has come from the Artic Network. Big kudos to Josh Quick and Nick Lohman in the UK for this kind of stuff. You should definitely check that out if you're going to get into Nanopore sequencing. Check out Artic. Okay, so when we talk about this sequencing, we have to do make some amplicons. So across a 30 kb genome, there's been a number of different primer sizes, and then that gives you a different type of number of primers. So the original Artic one has 400 base pairs, and there's 96 of those primers. And then there's also from John Sebastian Eden at Westmead, this 2.5 kb. I think that was referred to as the New South Wales primers in one of the previous talks. So there's 14 of those primers. But also in New Zealand, they've been trying out some different primer sets, 1.2, 1.5, and 2 with these numbers. Now you can find all these kind of protocols and protocols.io. So we've tried the Artic, and we've tried John Sebastian Eden's ones at the 2.5 kb amplicons. Then you can do a number of barcoding for multiplexing. So we've done with no barcoding when we were first testing. We've used the native kit. So you can have 12 barcodes or up to 24 barcodes. With the expansion pack, you can also do rapid barcoding, although this isn't necessarily supported by the Artic protocol. You can do it, and it reduces some of the steps needed for the barcoding. And if anyone wants to have a crack at direct RNA and barcode it, let us know. We've got some barcodes that can work. Now, to get this stuff to work for both the Artic pipeline, which I'll get into in a minute, and Rampart, which is live monitoring, you have to edit a few files to get them to work depending on the primer scheme you're working with. So these genomes and reference files don't really need to change, but it's just this schema here. It's just the bed file, which I'll show you in a second, and the primer JSON, which is just the coordinates of all your primers. So if you go into those, you've got your bed file here, which has the positions of your primers, the start and stop of your AB. So this is your 14 primers. And then you just do the same thing here with the positions here. Now this one here is really important if you want to do the live sequencing. And then this one here is really important for doing the Artic protocol. And you can actually get the instructions on how to do these modifications and how to run all of this at this GitHub here. Just look up Cypher PSY-FER, and you'll be able to find it. So when the Artic primers came out, the 96, the V1, there are a few, as was mentioned before, there were a few dropout regions. They've now updated to version 2, then version 3, and it's cleared all of these up. So this, obviously, you can see this one didn't have any barcoding because we've got, close to 20,000 times coverage. So that was on a MinION flow cell, bit overkill. And by the way, this the screenshot's coming from the live monitoring that you get from Rampart from James Hadfield, which is an absolutely fantastic piece of software that I think anyone doing nanopore sequencing should be using, because it's a really quick way to look at your samples, see if there's any dropout in your primers. Now, one of the advantages of using the 96 primer set is if your RNA is slightly degraded and you do get dropout in a primer, you're only missing a really small amount of the virus, of the genome. So there's a really good advantage there if your samples aren't super good. But if you're using the, say, 14 primer set like this, you get really nice coverage, and it's a little bit simpler to do. So this is it shows in the Rampart output, you can see the length distribution, so around 2.5 here, and these are the different amplicons, and where they overlap, you can see these little spikes here. So this was just near the start of a run at an average around 50 times coverage, but normally we get around 200 times coverage for everything. So this is Igor here loading up a MinION flow cell. But typically we run all of our sequencing on this flow cell here called a flongle. This is what a flongle looks like in real life. And we can put up to 12 samples on there with the native barcoding kit or the rapid barcoding kit. And if you've got one of these, my old boss, Martin, he's been sequencing some coronavirus sequences in his living room, which is just mind-blowing that you can do that. Once you get that sequencing going, you can spin up Rampart, and as it goes, you get this kind of overview of everything going on, and then it has all your samples. So again, this is one that wasn't barcoded, it was just a single sample where we're testing things out. So when you actually start multiplexing, you can have multiple samples. You get all your coverage statistics. So this is one of our runs from our collaborators at Kirby. And as you can see, pretty quickly you get 10 times coverage. These are like coverage maps over time. And I think it's about 4 hours or so. We had over 100 times coverage across all amplicons. And that's when you can start. You could probably easily get your results from there. So you could stop the flow cell, wash it, and then reuse it again for another run later on. So then once you get into the pipeline side of things on Artic, they both go through this length filtering and quality filtering of the FASTQ files. If you've barcoded your samples, then you have to demultiplex them. They used to just do pore chop. Now they're saying you should use the Guppy barcoder method. So if you don't have that, you could download that from the Oxford Nanopore website. Now, if you use the rapid kit, you have to use a few different commands to get the demultiplexing to work, because as well as when using Rampart as well. If you are interested in doing that, just check out the GitHub. It has instructions on how to do that as well. So once you've done the demultiplexing, you need to run choose either the Nanopolish pipeline or the Medaka pipeline, or if you're like us, we just did both to compare how good they go. So Nanopolish does works with the FAST5 files, the raw signal data, and it aligns the signal data to the bases so that you can then look for the probability of variance. So it indexes all the FAST5 files, does alignment with Minimap2, trimming, calls variants, merges those, and then does some filtering using the primer sites so that it kind of makes these depth masks where you get all this uneven coverage, and then creates the consensus just using VCF tools. Now, thankfully, coronavirus doesn't have a huge amount of like variability, so using a reference-based kind of assembly or variant detection is just really all you need. You don't need to do de novo assembly or anything like that. We've tried some of that. It works okay, but you can get some really messy stuff that comes out the other end. With Medaka, it's mostly the same, except it switches out nanopolish for Medaka, and then has this muscle step at the end. That's meant to say long shot. Whoops. The advantage here of using Medaka is that it only uses the FASTQ file, so you don't have to use the raw data, and that means that Medaka runs on average about twice as fast, or 30% faster than the Nanopolish pipeline. When it comes to results, we've compared, and they're pretty much identical. I think the only thing that we found was that Medaka was a little more sensitive to, if samples were a little bit dodgy or there was something strange going on. So it actually helped us solve some lab contamination problems. But I think from now on, we'll just be defaulting to Medaka, given that we don't have to cart around all the raw fast fight files. If anyone has any other comments on those differences and have tried things, I'd actually love to hear your experiences. So catch me up later in the breakout rooms. Now, in the original Artic pipeline, you meant to manually run each one of these in the command line for each barcode, but that's extremely tedious. So the way we do it is, you grab your list of samples and barcodes. So you say sample NCOV, 34 is barcode 7 or whatever, and so on and so forth. You point it towards your sequencing run folder on the gridiron that we were using, but you could do it on any system. You run it through a script, which essentially then just matches up the samples and barcodes, renames everything, and then for each one of those runs the Artic pipeline and sticks it in its own output folder. And a typical 12 barcode flongle run, it takes something like anywhere from 5 to 15 minutes, depending on if you're using Medaka or Nanopolish and how many reads you got. And again, you can check out how all that works on the GitHub to help you out. And if anything doesn't work, just create an issue. So the output looks like this. So Nanopolish and Medaka don't have the same outputs, but when it comes to the final, what you need to kind of have a manual inspection of the virus, as we said before, it's pretty good that it's only 30 KB because you can actually look at it on a nice long screen. So you're going to want this BAM file here. This is your VCF file with the variants, and then this is the consensus that's created with VCF tools that you would give off to GIS-AID, or to then go on to things like NextStrain, which I think is going to be talked about later on. With Medaka, same thing. You got the same sort of BAM, this pass VCF, and then this consensus file. You can see all the actual commands that are run manually inside these minion.log text files. So if you want to tweak the pipeline, you can actually see exactly what's being run. It's not just black magic, you can see what's going on. So I hope from that, it's pretty clear that you can use a variety of different primer lengths, and that should be updated, and then a number of different types of barcoding methodologies. You can monitor everything in real time, and Rampart is awesome. You should use it. That the Artic pipelines, I'm pretty sure Medaka is the superior one here, but they both give you the same outputs for further analysis. And if you're interested in doing Nanopore sequencing, please don't be afraid to reach out. It's been pretty amazing watching the entire international community come together and work together on a single goal. And I've had people from all these different countries asking me like how to get started, and I'm really glad that people have taken to this kind of thing really quickly, and I hope that continues after all of this. More collaboration and less competition. So with that, I'd like to thank the rest of the Genome Tech Lab, Ira, for getting all the collaborations up with Rowena and Bill, and Jill and Igor. You know, they're the stars here. They actually did the sequencing. I just pressed keys on a keyboard from my home in my dressing gown. And then Hasindu here, who's, you'll probably see some news from him soon, especially at the London Calling. He's got the entire Artic network running on mobile phones. And he's got this kind of grid computing on mobile phones, which is amazing. So look out for that.